Hello and welcome to Educated STEM. I'm Kate, the founder of Educated STEM, and I'm joined again today by my good friend and collaborator, Dr. Mahadeo Sukhai. And today we're going to dive into his own initiative, which is called Idea STEM. Please check back. We have a video where we talk about that previously. The link is above. But today I'm going to ask him about the tagline, "Inclusive science is good science." So can you tell me a bit about that, please, Mahadeo?
So first of all, what is good science, Kate? Good science is science where we feel like the question that we are posing is well designed, the methods that we are taking, the approaches we're taking to collect data, and the data that we get will lead to an interpretation that allows us to answer or perhaps even just refute a hypothesis, right? And so I said this in the last video, if we don't think about all of the lived experiences that humanity is made up of, then we are going to find ourselves in a space where the answers that we are getting when we do the research that we do are answers that are limited in scope. And that's not it's not a criticism, it's just a statement. It answers if we think about an environment where we don't take into account biological sex differences, if we don't take into account gender differences, if we don't take into account lived experience with disability, if we don't take into account ethnicity or culture or language or geography or socioeconomic status, or whether you live in a remote setting versus a city, right? Then what ends up happening is we are actually missing out on information, right? We're missing out on people who are not able to participate in the study, or who are not invited to participate in the study. And when we do that, we're missing out on knowledge that we could gain to help us shape the world around us, right? So here's an interesting sort of case study. If you think about the language in the Diagnostics and Statistics Manual version 5 around the behaviors that are diagnostic of autism, right? Now you think about the language that's there, and then you think about how that language is applied in different parts of the world. Interestingly, in a country like Japan, the usage of DSM-5 actually leads to a very significantly higher diagnostic rate of autism than in North America. Do you know why?
I've never heard of this, so no.
It's because the Japanese cultural conventions look a lot like the behavioral diagnostic criteria for autism.
Oh.
There was a paper that actually came out about this a couple of years ago that was highlighted on LinkedIn back in August, and it was a very fascinating read, because it actually suggested that that in thinking about something that is behaviorally defined, the authors of the DSM-5 neglected to take into consideration cultural conventions, right?
So we're saying that because of that, more people would be thought of as autistic in Japan, but that's not true.
Because the cultural conventions of the country mirror in some ways the diagnostic criteria, right?
So they need to go back and change
well, yeah,
DSM-5.
right? That's right. So here's a different example. So take eye contact, right? The cultural conventions of eye contact in North America and Western Europe are very much, if I look you in the eye, then I trust you and you can trust me, right? But there are places in the world where eye contact between two individuals is construed as a threat, not as openness and transparency, right? Autistic persons won't look you in the eye, and that's a neurological issue, not a lack of trust issue. I won't look you in the eye because, in addition to my lived experience with autism, I have nystagmus, and so looking you in the eye gives me a migraine, right? But if you're not looking somebody in the eye in North America and Western Europe, then you are not trustworthy, right? And so we map these cultural conventions onto things. We map these frames of reference onto things, and science is absolutely no exception to any of this, right? And so humans do science. Robots help us do science, but humans do science. And there's this really interesting thought process that goes, I'm a scientist, and my scientific identity says that I cannot be biased. Therefore, I am not biased because I'm a scientist, right? And the problem with that is you absolutely are biased, and your bias is that you're a scientist, and therefore you can't have any biases, right? And so if you acknowledge that you are biased by saying you have no biases, right, then all of a sudden you recognize your humanity, and you recognize that as a human being you have all sorts of biases, right? And the funny thing is, we teach people in science about how science has to be unbiased, right? And the scientific method is designed to be an unbiased approach to the gathering of knowledge, right? But that doesn't entirely work, because humans are the ones who are doing the data collection, and humans are the ones who are asking the questions, and humans are the ones who are interpreting the data. And so humans are absolutely biased, right? And so the important thing isn't that we're unbiased as scientists. The important thing is to recognize the limitations that our biases impose upon us. And then by acknowledging our biases and recognizing those limitations, we can then say, how do we correct for that? Right? So in the previous video, I gave sort of 3 examples, right, of scenarios where, in the context of the doing of science, a bias got introduced somewhere, right? And those examples were all things that actually can happen, actually have happened. And those are also scenarios where the folks who are supposed to sort of prevent the accumulation of biases never caught the bias. And in fact, in one specific example, the folks who are supposed to prevent the accumulation of biases actually introduced bias of their own accord, because they thought, intersecting social identity information was privileged data and shouldn't be collected, right? And so I come back to the tagline: inclusive science is good science, right? And what that effectively means is if we want to think about how to ask questions in such a way so that we're recognizing our biases and doing our best to eliminate them, then what we're doing is we're calling folks to the table. We're calling folks into our research teams who have lived experiences, who have experience, who have expertise, who have thought processes that are different than ours, that help us recognize the biases within ourselves and within each other. And by recognizing those biases within ourselves and within each other as a team, we can then say, let's ask the broader, more comprehensive, more complete question. Or if we cannot ask the broader, more comprehensive, more complete question, let us acknowledge that that is what we're actually doing, right? And so in doing that, then we scope our questions better, we scope our data collection better, we scope our data analysis and interpretation better. And all of that stems, no pun intended, from being more inclusive and more intentionally inclusive about who's on the team and how we're setting those questions up in the first place, right? That is why inclusive science is good science, right? We make the mistake collectively of thinking that all of these checks and balances are built in to the Western scientific method upfront. And while they might be, theoretically, we don't always apply them, right? And so we fall into that trap of, well, this is the way it's supposed to be by default. And if we don't change it, it's always going to be that way. But humans do science, and humans have biases all the time. Humans make mistakes all the time. And the interesting thing is, if we make a mistake that's in that zone of unconscious incompetence, we never know that we made a mistake, right? So inclusive science is good science.
Yeah, I'd agree with that.
That's why
Okay,
it's our tagline.
it's a good tagline.
Well, but I'm gonna just dive into this for a little bit longer, right? So
Okay.
think about how we teach science to our students. Think about how we teach science in elementary school. Think about how we teach science in high school. Think about how we teach science when we go to post-secondary, right? We teach science in such a way so that we always tell the outcome and strip away the journey, right? And so science loses its character, science loses its narrative, and science simply becomes a recitation of observations leading to conclusions, right? We don't learn in elementary school science or high school science, and we only start to learn in post-secondary science, that science is 90% perspiration and 10% inspiration. We only learn late in our learning of science that science doesn't always work, right? Do you know how many master students I have mentored who have come to the lab expecting every single experiment to work? Do you know what happened when I used to, for a friend of mine, actually run a lab module where I very deliberately set the lab module up to fail?
Ooh, you meanie.
Was it me? Or was it that that students, yes, they need to write a lab report, but the thing is that troubleshooting is a valuable life skill. You can't troubleshoot something if it works all the time, Kate.
That is true. But I can just imagine the master students thinking whether the experiment, I'm assuming that they assumed that the experiment will work
Yes.
because you have set the assignment. So it's something that should be reproducible, should be fine. And it
Well, no,
probably,
I didn't set it up that way. I very deliberately set it up to fail.
but I don't think they would have realized
No.
that. I can just imagine them wondering what they did wrong. Did
Yep.
they follow the protocol correctly? Did they add the wrong thing? Did they do something? So I can just imagine them sat there thinking, oh my gosh, what have I done wrong? As opposed to Mahadeo has deliberately designed an experiment that won't work.
But hold on a moment. Let's unpack that, because I had a scenario in my PhD where we had a mouse model of leukemia, and we phenotyped the mouse model of leukemia pretty quickly. And then the second stage of the PhD was to crossbreed the mouse model, introduce a second transgene, and voila, the leukemia should have happened within 2 months. That was the hypothesis. And you know what? The leukemia never happened.
Mhm.
Right? And we ran every conceivable test, and we validated that yes, the transgenic cross had been successful, and yes, we were breeding what we should have been breeding, and it was a pretty complicated cross, but it was working the way it was supposed to. And once we ensured that it was working the way it was supposed to, the only logical conclusion that was left was that the mice never got leukemia. Right? Then we went back and ran studies on the original mouse to make sure we didn't hallucinate the leukemia in the first place. Right? And at the end of the day, having done everything correctly, we still felt that we had done something wrong, as opposed to the other interpretation, which we came to only a little bit later, which was maybe instead of doing the thing that we expected to happen, the introduction of the transgene did the exact opposite. Right? But once we came to that conclusion, then we still had to justify it to my PhD supervisor and my thesis advisory committee, who didn't believe us.
I mean, this is true, and it mirrors something that I experienced in my first postdoc. So my supervisor had read a paper where something had happened, I want to say in yeast. And then he was like, great, let's do it in mammalian cells. This is going to be dead easy. We're going to have exactly the same result in mammalian cells. And that's going to be, I want to say the yeast paper was like it was a high prestigious paper. So he was like, we're going to get another great big paper. And I did it, and he kept having me repeat these awful homologous recombination assays, which took a long time, and it was just no fun.
That's right.
And he just kept saying, "You're doing it wrong. Like this is your fault. This is you." And then he went to a conference and saw someone presenting my data on a poster, and it was only once he saw someone else had the same information. And by that point, I had moved on to a second postdoc, so I hadn't published. And I got this panicked email from him saying, "Kate, you're correct. I just saw your data on a poster. We're going to get scooped. Write the paper immediately." And it was like, oh my gosh, you've got to be kidding me.
But I think that our stories have an extra piece of similarity, because it was only when another group based in France had actually published with a different version of with a different mouse model of the same leukemia, the same result that we were getting for 3 years, that I finally actually broke the logjam with my supervisor and got the thing published.
Yes.
Right.
But I think sometimes they don't want to believe that the hypothesis is wrong.
But the whole point of hypothesis testing, and you learn this if you take second-year statistics, is that no hypothesis can ever be verified. They can only be falsified. Right? And so the whole point of a hypothesis is to prove the damn thing wrong. Right? That is the purpose of generating a hypothesis. It is to prove it wrong. Right? And so because of how we set science up and how we set up the expectation around science, I think we do ourselves a fundamental disservice, right? Because we train whole generations of scientists that things have to work all the time, right? And we also train whole generations of scientists on their intellectual invulnerability as scientists. And so we don't question the voids in our knowledge. We don't question what we don't know that we don't know. Right? Go back to the group that was doing the work on aging in place and older adults and assistive technology use. They had not fundamentally thought to check whether their survey could interact with assistive technology. That is a ginormous gap, given the nature of their research question. Right? There's another kind of story that I can share that I was not part of, but I did sort of encounter this. So there was a medical device related to diabetes that had received regulatory approval in Canada with a disclaimer that the device was not safe for blind or low vision persons to use. And at that point in time, I was substantially concerned that was actually written there as a research lead for an organization that worked in that space, and so I was curious, why did the regulatory agency make that disclaimer? They literally said, this is not safe for blind low vision persons to use, and they cited a scientific reference. I went and I looked up the paper, and the paper in the published version of the manuscript said nothing about persons with vision loss. And you actually had to dig in the supplemental methods to find the one sentence that the regulatory agency actually referred to. And the sentence basically said, "We did not know how to make a usability test accessible to blind folk. Therefore, this device was not tested on that population." Right? Now, here's a case where the authors identified that they didn't know something, but they did not know that it was actually pig simple to correct the issue, because there's a few of us who know how to do accessible surveys really well, and none of us got a phone call. Right? And so, this is what we want to think about when we talk about how we teach science. It isn't just about, I want to teach science inclusively. It's about, how do I teach science so that fundamentally I build in the tenets of being inclusive into the design of the research question, right? How do I understand that science is a journey, and it's a journey undertaken by human beings? And that journey needs to be one that is holistically recognized, and we are actually paying attention to things that we wouldn't pay attention to otherwise. That's the whole point of science, right? We can't discover things if we never ask the right questions.
Thank you for joining me today, Mahadeo. The next time we meet, we're going to talk about how do you do science through DEI or IDEA. So please join us when we talk about that. Please like and subscribe to the channel, mine and over at IDEA STEM. And I look forward to seeing you next time. Thank you. Bye-bye.